This is Bio 181, Chapter 14 on DNA structure. Okay, so we're going to look at the building blocks of DNA. So it's made up of repeating subunits. These subunits are called monomers. And in the case of DNA or RNA, which are nucleic acids, these are called nucleotides. Nucleotides. So each of these that are connected with a covalent bond are nucleotides. So the specific structure of each nucleotide for DNA is going to consist of a phosphate group, which we can represent with PI. It is actually a functional group we are familiar with, PO4 2-, so it has a negative charge of 2 on it. It's got a phosphate. It's what we know and love from ATP. In addition to that phosphate, we also have a sugar. So in the case of DNA, it's going to be called deoxyribonucleic acid. It is a pentose, which means it has 5 carbons on it. 1 carbon, 2 carbons, 3 carbons, 4 carbons, 5 carbons. Okay. So DNA, it is called deoxyribose, OSE on the end, means it's a sugar. And then that is connected to a nitrogenous base. Nitrogenous base. And I just usually draw this as kind of a rectangle. There's a few different forms. In the case of DNA, there are 4 options for nitrogenous bases: adenine, guanine, cytosine, and thymine. Okay, those are written up here, and you do need to know those. In the case of RNA, which we'll get to, the difference is in RNA, instead of deoxyribose, in RNA, the sugar is called ribose, so it's simpler. And that's shown over here. The only difference between these 2 sugar molecules is the presence or absence of an oxygen. And then for RNA, the bases are quite similar. We have adenine, guanine, cytosine, but then instead of thymine, we have a base called uracil. So that's really the big difference here. Okay. This is one side of a double-stranded DNA molecule. So if we look at one side, you can see there are multiple nucleotides that are hooked together here. Here's one nucleotide. We've got our phosphate group. We've got our sugar, which ends in OSE, our deoxyribose, and then we've got our nitrogenous base. Okay. And there are multiple of these put together to create a polymer. The bond that is holding them together is called a phosphodiester bond. And that's just the name of this bond shown here, which links together the nucleotides in the sequence. You'll often hear the DNA backbone referred to as a sugar phosphate backbone. Sugar, here's my 5-carbon sugar, here's my phosphate, okay? Phosphate, and it's just repeated over and over again. So it's a sugar phosphate backbone, and then the bases are held out to the side. One other thing to call your attention to is the numbering system for the sugar. So if I look at this, here's my sugar, and the carbons are numbered. This is going to be important for determining the orientation of the DNA. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Again, this is a sugar. It ends in OSE. It's a 5-carbon sugar. In the case of DNA, it's deoxyribose. So when we talk about the strand, the fact that this carbon down here is the closest to this end at the bottom in the screen means that this end is called the 3' end. the number 5 carbon is closest to the opposite end. So this end is called the 5' end. So DNA consists of 2 anti-parallel strands. So that means one is going to be oriented in the 3' to 5' end. Whoops. This is the 5', here's the 3'. Okay. And the other strand is going to be in the opposite direction. So it's going to be in the 3' to 5'. So that's anti-parallel. They're held together by hydrogen bonds that link the base pairs. So between cytosine and guanine, they're always going to be together. And adenine and thymine are always going to be linked together. They're linked together with hydrogen bonds. If you remember, hydrogen bonds are represented with either dashed or dotted lines. So there's three hydrogen bonds that link together cytosine and guanine. And then there's 2 hydrogen bonds that link together adenine and thymine. Okay, so 2 hydrogen bonds. Hydrogen bonds are not as strong as covalent bonds. Remember, covalent bonds, they're sharing electrons. In hydrogen bonds, it's just partial positive hydrogens that are attracted to the partial negative charges in other electronegative atoms. So it can easily be split if needed. This looks busy, but we've just looked at the building blocks. So you can see up here, here's a nucleotide, upper left, right? Below it, another nucleotide. Each nucleotide consists of a phosphate group, a sugar, and a nitrogenous base, right? They're linked together with the phosphodiester bonds, okay? It's a sugar phosphate backbone. So sugar, phosphate, sugar, phosphate, sugar, phosphate. It's double-stranded. One strand's over here on the left. The second strand is over here on the right, right? And they're held together with these hydrogen bonds that are found between the bases. And again, you can see 2 hydrogen bonds between adenine and thymine. A and T, you can think about, they're always together, apples in the trees. So here's my apple, here's my tree. Okay, so adenine and thymine always together. C and G, you can think about the car and the garage. So you've got your car, right? And then you've got it in your garage. Okay, so that might help you just remember that pairing. All right, that's it for the structure.